how did you get into this area that we know you for?
Yeah, well, thank you. Well, right now I'm a professor of biology at Boston College. I teach graduate students. I have an active research lab with professional scientists and technicians. I teach general biology to the non-majors and also cancer metabolism to the advanced undergraduates in the biology program. So I have a broad background, but over the many years that I've been here at BC, 39 years now, I've taught neurobiology, neurochemistry, and a variety of other subjects. I worked for many years in the field of epilepsy, which started when I was a postdoc and then a faculty member in the Department of Neurology at Yale University in New Haven, Connecticut. So I did a lot of work on epilepsy, mapped genes in mice, which would hope to lead to better management of epilepsy. But I did most of that at Yale because everybody was interested in that kind of stuff. But when we came up to BC, one of my students said, "Oh, you should do the ketogenic diet for epilepsy. It really works." And I said, "No." And when I was at Yale, I tried to write an internal grant on ketogenic diet for epilepsy and the animal models that I had. And they said, "Oh, no, nobody cares about ketogenic diets for epilepsy." Are you kidding me? I said, "Now it's one of the top non-toxicological management, non-pharmacological management of cancer that really works." Interesting.
The seizures.
For managing epilepsy. And to this day, we're really not sure about the multiple mechanisms by which ketogenic diets work for managing epilepsy seizures, recurrent seizures. It was started in 1921 by Wilder at the Mayo Clinic. And what we found here at Boston College was that it had a lot to do with the levels of blood sugar. We could keep mice and people managed in their seizures for quite a while, but even a slight elevation in blood sugar from whatever within a short period of time would trigger an epileptic seizure. So it wasn't clear whether it was the elevation of ketones or the management of low sugar that was responsible for maintaining a non-epileptic or a non-ictal, let's put it that way, event in the brain. And to this day, we're really not sure how all this is stopping. I mean, there's a lot of hypotheses, but it's still not clear.
Well, this makes sense to me a little more now that you had that background with the ketogenic diet and epilepsy, then applying that more to cancer. Have you kept up with the research on seizures? And there seem to be a subset who have GLUT4 deficiency, right?
Yeah,
glucose in the brain. But generally, everyone gets a little bit better, or 2/3 get better if you have seizures and you go on a keto diet. And still today, it's not well known in the epilepsy world because they're mostly drug focused, right?
Yes.
That's a theme in medical care today in general. But that's really, it's the puzzle is coming together a little bit. How did you pivot then to other types of? We
realized the work, the way that ketogenic diet seemed to be linked to blocking seizures was having low blood sugar and elevated ketones. And when we put mice that had epilepsy on calorie-restricted diets, where blood sugar would go down and ketones would go up naturally without a ketogenic diet, we got the same management of seizures. So it was clearly a ratio of glucose to ketones that was blocking epileptic seizures, but the molecular mechanism by which that happens in neural circuitry is still debatable, whether it's an increase in inhibitory neurotransmitters, a reduction in excitatory neurotransmitters, or the signaling pathways that are involved is still up for debate. But we had a lot of biochemistry on brain cancer at the same time. We were looking at glycolipid metabolism in brain tumors. I had a number of mice that were built with brain tumors, and we were always asking whether the glycolipid patterns in mouse brain tumors were similar to that in human brain tumors. So I did some comparisons, and there were some similarities, but there were a lot of differences. And the main problem was it was hopelessly complicated whether you had a human brain tumor or a mouse brain tumor. It turns out that a lot of the blood vessels have different glycolipid patterns. So the angiogenesis, the blood vessel, the infiltrating immune cells all had their own unique ganglioside glycolipid patterns all mixed together. So it was very difficult to ferret out who was responsible for what. But it turned out that I was looking at glycosphingolipids, and a company gave me a drug. We were working on Tay-Sachs disease, which is a glycolipid storage disease. And we were looking at blockers of ganglioside biosynthesis. And like everything that comes into my lab, I always throw it on an epileptic mouse or a brain tumor mouse.
Well, if you have a hammer.
Yeah. That's absolutely right. Does this drug, if it doesn't work, so I put it on the epilepsy mice, this ganglioside inhibitor, and the seizures became reduced. I said, whoa, this is really exciting. So the drug company decided to give me a lot more money. And not only that, I put it on brain cancer, mice with brain tumors. And they got very excited because it was shrinking the brain tumors. And I said, wow, what is going on with this drug? This is really an unbelievable drug. It blocks epileptic seizures in mice that have genetic epilepsy, and it shrinks glioblastoma models in the mouse. So I said, whoa. So the company gave me a substantial amount of money to figure out what the mechanism was by which this drug was working. And, it was interesting when we fed the drug either to the mice with epilepsy or to the mice with the brain cancer, their body weights went down as if they were calorie restricted. And the epilepsy or the brain cancer, the tumor stopped growing or grew very, very slowly. The seizures gradually dissipated, but the animals lost body weight. And I was saying, what's going on with that? It was acting like a calorie restriction, because when you calorie restrict mice with brain tumors, also the tumor went down, shrunk. So I decided to put in a body weight control on this drug. So I would just feed the animals less food to match exactly the body weight of the drug. And sure enough, it was equally powerful in stopping seizures or shrinking the brain tumor. So I said, well, what is this drug doing? And it turns out the drug blocked sucrases in the gut. So the animals ate the food, but they couldn't digest it to get the carbohydrate out of the food. So the drug was causing an indirect calorie restriction. And that's the reason why it works so well.
If you want to learn how to implement the ketogenic diet and the Press Pulse protocol into your cancer management, just visit school.com/keto for cancer, the new community by our research analyst Johnny Rockermeyer. Within this new community platform, you can connect with other patients and learn from their experiences. You can also just search through the posts other members have already made. You will have access to our classroom section as well, which provides practical advice and tools for the best results. These sections cover all parts of the Press Pulse protocol that you should implement. Starting with the general scientific concepts by Dr. Thomas Seyfried, we will show you how to start the ketogenic diet, including which foods to choose and which to avoid. Additionally, we prepared a list of doctors specialized in metabolic therapy, as well as details about glutamine inhibition and hyperbaric oxygen protocols. So, take a look around at school.com/keto for cancer.
So when I told the company that this drug was working indirectly through calorie restriction, immediately all my support and funding disappeared completely.
Sometimes you don't want to be quite so honest. Yeah.
Oh, they dropped it like a hot potato. They didn't want. But the drug actually worked on ganglioside, was shrinking and that's what made them think that it was working. But it wasn't the gangliosides. We ruled that out. It was the calorie restriction that lowered the blood sugar. And then I started to think about how does this work? And then I found Otto Warburg's work from years and years ago, the great German scientist, and he was saying, we have a few. I'll play. You have a few minutes to unpack that. How did you kind of rediscover the Warburg effect? Well, because Linda Nebling at Case Western got her Ph.D. there in nursing, and she was giving little kids ketogenic diets that were hopeless cases. They were poisoned and irradiated and surgically mutilated, and these little kids with high grade gliomas were given up. And as a nursing student, she said, "Well, they got nothing to lose. Why don't we give them ketogenic diets? So that immediately brought us, because we knew about ketogenic diets and epilepsy, and we knew that the ketogenic diet worked because it was lowering glucose and elevating ketones. And she said she got dramatic results on these little kids that had high-grade glioma with ketogenic diet. So, and then then she said this might have something to do with what Otto Warburg said. So I said, well, I heard of Otto Warburg. Everybody's heard of this guy. What the hell did he do? So I went back and started to unpack all the work that he had done. And I said, man, this guy had it right. So then from that point on, from about the year 2000 up until present, so more than 25 years, I guess 25 years, actually late 90s, we started to look deeper into this cancer thing, looking at the metabolism.
So before we get there, the analogy is very interesting, because I kind of rediscovered the treatment of diabetes and obesity from 100 years ago, and it was a low carb diet. What did Otto Warburg actually do, and what did he learn? And but I know it's more complicated than that. But so you went back and read the articles and
So I said, well, Warburg clearly said back in and then from his work in the 1930s, goes back to actually the middle 20s, I guess, 1920s, all the way up to the 60s, that cancer was a metabolic disorder. And all cancer had chronic damage to oxidative phosphorylation, the ability to produce ATP with oxygen. And as a result, to get dysregulated growth, they had to compensate by switching to energy that involved no oxygen, like fermentation. Energy without oxygen is fermentation. And he said, in order to get cancer, you have to have chronic damage to the ability of the organelle, the mitochondria, to produce energy through oxygen, but it has to be compensated by that cell's same ability to upregulate a fermentation metabolism. So therefore, there's two steps to get cancer. Number one, chronic damage to oxidative phosphorylation. And step two, a compensatory substrate level phosphorylation, or what we call fermentation. And he clearly showed that all major cancers had a reduction in oxygen consumption and an excessive production of lactic acid, which is the end product of the glycolysis pathway, the Embden-Meyerhoff-Barnes pathway. So, and he said this was the characteristic of all cancers. And I read the papers very, very carefully in light of what I'm seeing for the first time in my own research. And I'm saying this guy was right. But he wasn't completely right. And I didn't realize at the beginning where he made some mistakes and why his critics misinterpreted a lot of what he said, and continue to this day, as you and I are speaking, continue to misunderstand what Warburg did and the mistakes that Warburg made. In those days, oxygen consumption was considered a direct linkage to ATP production through oxidative phosphorylation. So Warburg made this connection based on Hans Krebs's work of the Krebs cycle, that each mole of oxygen breathed produced 7 moles of ATP, which is the energy from oxidative phosphorylation. So he showed that all these cancer cells were breathing less. They were consuming less oxygen. And then they were all producing excessive amounts of lactic acid, which everybody knows that if you hold your breath or have a heart attack or whatever, lactic acid builds up in your bloodstream. Even sometimes with epileptic seizures, when the patient stops breathing during the seizure, lactic acid explodes in the bloodstream because your tissues are now deprived of oxygen. And in order to prevent themselves from dying, you immediately upregulate a powerful fermentation metabolism, which is energy without oxygen. But the waste product, the end product, is lactic acid. So you see lactic acid build up. And what Warburg said is all cancers have a deficiency of oxygen consumption and a buildup of lactic acid, and that's because they have a chronic damage to OXPHOS, which is compensated for by a lactic acid. He showed that even in 100% oxygen, all tumor cells continue to throw out lactic acid. And that could only happen if there was a chronic damage in their mitochondria and the ability to oxidative phosphorylation. Now, well, that was logical, and all cancers seem to have this, and they refer to it now as the Warburg effect, and everybody seems to be comfortable with that, although most people misinterpreted what it means. But so I looked at that, and I looked at all these different kinds of things. And what I found, I did things his attackers, the people who didn't agree with him, said that, there are some cancer cells which have completely normal oxygen consumption, and therefore Warburg must be wrong. And the reason why they continue to produce so much lactic acid is because they have a defect in lactic acid in glycolysis, not a defect in oxidative phosphorylation. And this argument is still today. Many people refuse to believe that Warburg was correct. So what I did, and my colleagues here, is we carefully examined oxygen consumption. Is it an accurate marker for ATP synthesis through oxidative phosphorylation? Is there a direct relationship? And the answer is only in normal cells and tissues is that a direct relationship, and not in cancer. And this is where Warburg and all of his critics made this big mistake. They assumed that the oxygen consumption in a cancer cell was producing ATP as efficiently as oxygen consumption in a normal cell. We and others have shown that the oxygen is consumed in cancer cells but not used entirely for ATP production. So it's a false. You're using it.
What else does it do? What is that?
They produce reactive oxygen species, ROS. So that a lot of the cancer cells take in oxygen, and they use it for the production of reactive oxygen species and a variety of other activities independent of ATP production through oxidative phosphorylation. So you can't use, in a cancer cell, you cannot use oxygen consumption as a reliable substitute for oxidative phosphorylation, as a reliable surrogate. This is the new stuff that we're presenting now as we speak in some of our new writings, which then throws the whole cancer field in a tiff, because now everybody was using oxygen consumption and saying OXPHOS is normal in cancer cells. Warburg was wrong. Warburg was right that mitochondria defective, his quantification using oxygen consumption was not correct. And to this day, it's not correct with most of the people that say Warburg was wrong. So we cleared that up. And then we also found out that the mitochondria in cancer cells produce ATP through a fermentation, amino acid fermentation. So the mitochondria in cancer cells can ferment. Nobody knew this. We have new evidence now that everybody knew glutamine was used by tumor cells in huge amounts, but they never knew that it was fermented. We have now direct evidence that glutamine is a fermentable amino acid. So when you look at a tumor cell, you say, see, it's consuming oxygen, and it's making ATP, making it look like it's normal, but the oxygen consumption is used for reactive oxygen species, and the ATP is coming from a glutamine fermentation metabolism. This throws the whole field into a. Now, they're going to come to know this, and they're going to come to know real soon, because Otto Warburg was not incorrect. He just did not have all the parts of the puzzle like we now have it. So therefore, cancer can grow without oxygen because it ferments, and it ferments glucose and glutamine, the amino acid. So we put this together, and now we know how to manage cancer. We know how to prevent it.
Well, I guess what is remember the telephone game where you would say something and someone would say to the next person, and then by the end of all this, what got to me, the Warburg effect, cancer like sugar. Cancer like sugar, and that isn't the whole story by any means. But to what degree now is that true? So you learned that a keto diet was something that children with epilepsy could have. It kind of probably made you more comfortable with the whole concept of not feeding animals and humans carbs, right? And then the idea that blocking the sugar absorption with a drug started having these effects even on a brain tumor, the glioma brain tumor model. How then did this alter or shift the science that you wanted to create and generate?
Yeah, well, that's an excellent point, which is still not understood by the majority of oncologists and is refused to be accepted by the cancer industry itself, that these tumor cells can't survive without fermentation. And the two fuels that ferment are glucose, the sugar, and the amino acid glutamine. And when they say, oh, cancer cells can switch to other amino acids, no, they can't. We tested them. None of them are as effective as glutamine. Oh, they can go to fatty acids, and no, they can't. And I threw it out. I said, challenge, you show me any tumor cell that can live in the absence of glucose and glutamine on fatty acids and can survive and not just for an hour. Let's show me several days this tumor cell, because normal cells can do this. Normal cells don't need glucose and glutamine. They can survive on other fuels. They can survive on ketone bodies and certain things like this. So and nobody can show that. So we know cancer cells are absolutely dependent on two fermentable fuels for survival. Very clear, although it's not accepted because they think that some gene mutation is involved. And we showed that all those gene mutations are downstream epiphenomena. They're not the cause, they're the effects of the abnormal energy metabolism. But let me get right to the core. Once you understand, just like the epilepsy, you can manage seizures if you lower glucose and elevate ketones. When you lower your blood sugar, whether it's through water-only fasting, through ketogenic diets, or low-carb diets, or however you want to speak about it, and we built the glucose ketone index calculator, which calculates the ratio of glucose to ketones in the blood. And when you have low GKI values, the tumors start to get hammered big time. They cannot adapt. They're locked into a fermentation metabolism dependent on two fuels. And then people say to me, well, you'll never get glucose to be as low as you need to kill the tumor cell. Oh, yes, you can. Absolutely. And if you remember, Eric, the study by Drinick, where he took the obese people, he published a paper, I think it was several very obese men, and he fasted them for 21 days, just water only, for 21 days, and then injected them with insulin, and brought the blood sugar was down to 0.5 millimolar, or 9 milligrams per deciliter. I mean, you as a physician most normal people would be dead. These guys were perfectly cognitive. They had
we don't recommend doing that at home.
No, we don't recommend doing it. But the study showed that there was no adverse effects from, and they attributed it because the ketone bodies were replacing glucose for the brain.
Keto adaptation, yeah.
Yes, keto adaptation allowed these guys to be resistant to very, very low glucose levels. Yeah. So you can.
If you're running your body on ketones, the glucose level doesn't have to be so high.
No.
So the puzzle today still is how to not allow glutamine to be around.
Yeah. So that's why we developed with my physician colleagues the press pulse therapeutic strategy, which has its linkage to paleobiology. What I do in my research is I look deeply into evolutionary biology and concepts of science. And paleobiologists were trying to figure out what caused great extinctions of organisms on the planet over the history of our Earth as a planet. And these massive extinctions seem to happen when two unlikely events happen together. A chronic stress on large numbers of organisms on the planet, killing off the weak ones, but many of the strong ones were surviving. Then some sort of an impact from a comet or an explosion of volcanoes all over the Earth was the pulse. So the chronic environmental stress together with some pulse caused complete exterminations of almost all organisms. So I developed that concept for managing cancer. I said, if we can lower the blood sugar and put tremendous stress on the cancer cells, but a few of them that are surviving on the low glutamine, then we come in with a drug that just pulses the glutamine, short pulses, which wipes out large numbers of cancer cells. Because we also know, based on our research and the research of others, that our immune system needs glutamine to function. Our gut needs glutamine to function. Our urea cycle needs glutamine to function appropriately. So if we stress too much on the glutamine, we cause harm to the body. But if we come in short with pulsing while we're keeping the chokehold on the glucose with the press, the diet can press the glucose down massively. So we come in with short pulses to target the glutamine, keeping the gut healthy and the immune system healthy. We can then, our normal healthy immune system gets paralyzed but not killed by glutamine targeting. So what happens is when we pull off the glutamine targeting, our immune system comes in and picks up the dead cancer cells and then gets rid of them. And then we come back and we pelts it again. So it's a press pulse concept that gradually degrades the tumor while allowing the normal functions of the body to work without paralyzing the normal functions of the body to eliminate the tumor. It's a beauty. You have to know human systems physiology, biochemistry, evolutionary biology, and genetics. You put all that together and you can come up with a nice non-toxic therapy for managing cancer. Doesn't require all these toxic drugs, hunting for genes, which are all downstream epiphenomena. So once you understand the biology of the problem, you can then design non-toxic therapeutic strategies. And those folks that do it and understand how it works, you can't believe the therapeutic efficacy of these people. Cancers go into remission. So I'm not saying we cure cancer, but we certainly can manage it for long periods of time without toxicity. And maybe there are some resolutions.
Let me just see if I have that. So you use some kind of diet to get the glucose low, and then what's the pulse again? Do you take away glutamine?
Yeah, no, okay. So glutamine is a non-essential amino acid. It's made from glucose. So if I'm lowering glucose, I'm also not producing as much glutamine by lowering the glucose. Okay. But that's that. But we also know it's extremely important for physiology. So I come in with glutamine targeting drugs. Okay.
Okay.
Yeah, and these drugs like 6-deoxynorlucine Don, which was not available to people, it's a very powerful, it should be. When they have all these crazy drugs out there that cause your hair to fall out, your gums to bleed, diarrhea, nausea, they say these are the drugs they say well you should take, but the very drug that works the best is not available for you to take. Can you believe this?
Yeah, has that changed? Has that changed? I remember talking to you a few years ago.
Yeah.
One of my patients, are they still not available?
No, they're not available,
Okay.
Which is crazy. Why the Food and Drug Administration allows all these toxic poisons to be allowed to treat cancer patients? And
the money.
Yeah, but I don't know if I know that, but I don't think the Food and Drug Administration are getting paid off by the pharmaceutical industry. Are they? Maybe they are, I don't know.
No, but it takes a lot of money to generate the studies that would be needed, I think. But
yeah, so we've already done the studies, and the studies were done. Don't forget, Don was used in little kids with leukemias, and Don was used in the past, and it had some therapeutic benefit, but not for everybody. They didn't target the glucose when they were using the Don. It's not going to work. It's not going to work the way it should work. Again,
well, I wonder. So, what would you need? A lot of people will watch this who have other day jobs that might be helpful. So there's a drug that's available. You can purchase it from a drug company, but it's not approved for human use.
Yes, not approved for human use.
So you get in some hot water because you're not an MD, right?
But well, listen, Eric, if I had cancer, I would buy it from the chemical company and use it because I've seen how it works.
So I remember some years ago I worked with the inventor of the nicotine patch.
Yeah,
and I was asked to get drugs which then would be prepared by our university pharmacy for the human use. They make sure there's no mold and all of this.
Yeah.
And so under some sort of investigational drug research, this could be done and changed into human use. But I would like to, it would be wonderful if young oncologists watching this who want to try a treatment that you've done now, and tell me how many years or how many models of cancer you've used this press pulse therapy in?
Well, we did it in mice first, but now when humans try it, they do much better than the mice. Our basal metabolic rate is 7 times slower than that of the mouse. So our bodies can deal with this so much. And then you have a physician who's actually looking at the patient and moves the patient as to how you feel today. The mouse can't tell us that. So, right? So we're the success of ketogenic metabolic therapy for humans is so much more powerful than it is in the mouse, because we look at the mouse and, obviously if he's breathing heavy, he's not moving, his hair looks kind of disheveled, we know he's not feeling so well. And we try to move him away from those states, but we can't ask it. We can only know by the way he eats and the way he moves around that he's okay.
So you are working with in any research setting or with doctors who are using this with people?
Yeah, well, no one's using Dawn. There might be a few people actually who, the wealthy people can get whatever they want. If you have a lot of money, you just go out and buy it, you pay a physician, give it to me, and here's the dosage because it's already been published in the scientific literature. So, it's been used on people, and evidence is there. How much should I use? When should I use it? It's all been there.
But I would still like some young oncologist or old. A lot of times there are people at the end of their careers who haven't fixed anything, and they say, oh, well, we'll let one of our postdocs look at a keto diet.
Yeah.
What can that do? Now there's a whole neurodegenerative field based on what seems to me that those Zhang Rou and Adrian Schecht seemed like they were at the end of their let's let those postdocs do what they want, and now they're stuck on this field.
Oh yeah, well it works, but the thing of it is you've got to know the tools that you have and how to use them. And I find that the oncology, like for example, in medical training, no one speaks about metabolic therapy as the future for managing cancer. They're all indoctrinated to think cancer is a genetic disease. And they're going to find these drugs that target your ALK mutation, your EGFR receptor, or these crazy things that are all downstream epiphenomena. I published a major paper showing that cancer is a mitochondrial metabolic disorder. It's not a genetic disorder.
Tell me about this study. It was brilliant. Tell me about this.
Well, it was several studies that I put all the stuff together, and I said you have to be cognitively challenged to think that cancer is a genetic disease. Either that, or you have to be a hopeless ideologue. Anybody that would have a few functional brain cells and would read the papers would have to realize cancer is not a genetic disease. So what I did was I went through a lot of different things. And where did this idea come from in the first place that cancer was a genetic disease? It came from Theodore Boveri in 1914, who studied sea urchins, and he had a hunch that cancer might involve some chromosomal abnormalities, but he said, I'm probably completely wrong. I'm an interloper. I don't know anything about cancer. And yet the field brings him as the founding father of the gene theory. But what I found is that if you look at some of the papers, like Sir Michael Stratton, who is very well known in the field of cancer genetics, he found several breast cancers that had no mutations. How can you say in the somatic mutation theory that cancer is a genetic disease if I find cancer cells that have no mutations? How do you explain that? How can you say that? And what I find is happening? And also Parsons with glioblastoma found a patient that had none of the mutations, no mutations in what they predicted. So, well, how do they do it? Guess what they do? They ignore it. They don't say anything about it. They absolutely ignore it. I said, what do you mean? You have direct evidence here saying these cancers are not due to mutations, and yet you're saying cancer is a genetic disease. How could you say something like that? And the National Cancer Institute says right on their homepage there, cancer is a genetic disease consisting of 100 different diseases, all different genetic mutations. Nothing could be further from the truth. don't these people at the NCI read the scientific literature? So let me go on. Eric,
how did you convince yourself? How did you convince yourself that it's not?
Well, then I'm going through the list, and now we find that not all mutations are bad. Only the driver mutations are the ones that cause dysregulated cell growth. Now we're finding new papers coming out that show driver mutations in our normal tissues that never become cancer. So that throws cold water. All of those are suggestions that maybe cancer is not a genetic disease. But when you go back and look at the nuclear mitochondria transfer experiments, that's the nail in the coffin, where scientists took Israel and Seyfried from Vermont, took the nucleus of a tumor cell and put it in the cytoplasm of a normal cell, and they got complete regulated growth despite the continued mutations in the nucleus. And when they did the reversed experiment, where they took the nucleus of a normal cell and put it in the cytoplasm of a tumor cell, they got dysregulated cell growth and cancer. And this was done in frogs, in mice, that was done in vivo, in vitro, repeated over and over again by some of the best developmental biologists in the field. None of them were testing Warburg's theory. They were just asking how far a cancer nucleus could drive normal development. And in each case, it drove it to a certain extent level, and then the whole system aborted because the mutations in the nucleus were aborting development. But at no time did those mutations lead to dysregulated cell growth, which is the definition of what cancer is. So when you look at the transfer experiments and all these others, you have to be cognitively challenged to think that cancer is a genetic disease.
Well, so to the non-scientist listening, the genetic material is in the nucleus.
Yeah.
Right. So if you're going to argue that the cancer is entirely genetic and you're changing the nucleus, which has the genetic material to other cells, this really does throw a wet blanket on that idea. And then if you change the mitochondria from cell to cell,
you can shut down. So it's very interesting. All these oncogenes and tumor, these things that people think are important, when you put new mitochondria into the cytoplasm, they all turn off because you don't need to ferment anymore. Because the reason those oncogenes are facilitators of fermentation metabolism. So when a cancer cell becomes dysregulated, it has to have all of this glucose and glutamine come pouring into the cell to replace oxidative phosphorylation, oxphos. So you have to open the floodgates to get in a poorly used fuel because you're wasting so much of it as waste product. So they are driven by oncogenes. But if you put a new mitochondria in there it's like putting a new engine in the car. Boom, everything's working again. You turn off the oncogenes are all turned off. So it's very clear what's going on here. So
something you said earlier, if you could just explain, so that the genetic mutation is an epiphenomenon. Explain that to the general public.
Okay. So, when you look at cancer, yes, they're full of mutations. All cancer cells have loaded with, and now over millions now. This is the big genome projects have identified millions of mutations, right? Where do all these millions of mutations come from in these cancer cells? And they come from
radiation.
Radiation can do that. Chemical carcinogens can do that. But how do chemical carcinogens cause mutations in the nucleus? They base their studies on salmonella, which is a bacteria. So you put chemical carcinogen on the bacteria, and you get mutations. But in humans, the chemical carcinogen goes into the mitochondria, and the oxygen that we breathe forms oxygen radicals called ROS, reactive oxygen species, ROS. This is the superoxide anion and the hyperoxygen molecule. They damage DNA, proteins, and lipids. So the mutations that we see in the nucleus are downstream effects of the mutagenic process of the mitochondria. So bad mitochondria that don't function properly produce ROS that are carcinogenic and mutagenic as a secondary effect of the damage to oxidative phosphorylation. So our nucleus collects all these mutations, and almost the entire cancer field is focused on all these mutations that are all downstream epiphenomena. So that's why you have Keytruda, Opdivo, And you have all these immunotherapies that you hear because they're trying to allow the immune system to function despite all these different kinds of mutations. So most of the therapies, now, here's the situation. The immunotherapies, CAR T immunotherapy, Opdivo, PD-L1, PD-1 inhibitors, and all these things you hear about advertised on TV at night, how you're going to do so wonderful. They're all based on the somatic mutation, the gene theory of cancer. If the gene theory of cancer is not correct, you're not going to make any major drop. Listen, Eric, we have almost 1,700 people a day in this country dying from cancer, which comes to about 70 people an hour, and it gets worse and worse and worse every year. And every year we hear new drug, new therapy, all based on the somatic mutation theory. So if the somatic mutation theory is not the correct theory, you're not going to get optimal outcome any more than the geocentric theory could explain the movements of the planets. Copernicus came along with Kepler and Galileo and said, no, the Earth moves and it circles the sun. And once that happened, everybody, oh, yeah, But for 1,800 years, people thought the Earth was immovable and the sun revolved around the Earth. Right now, the cancer industry thinks that the nuclear gene mutations are the drivers of the dysregulated cell growth. No. They're the effects. They're not the drivers. They're the effects.
So how, if we could only transplant the mitochondria in every cell in the cancer tumor, or how do you fix the mitochondria, cut the sugar down? Is there any way to fix that mitochondria?
Well, that's the future. I have been approached by entrepreneurs or these guys that say the future of cancer is mitochondrial transplantation. But that's not going to be in my lifetime. So I'm not going to be around long enough to do mitochondrial transplant. But yes, that would, in fact, in culture, that was shown. If you can put new mitochondria in the cells, you can bring them back to growth regulation. I'm alive long enough to let's kill the tumor cells without toxicity so that we can also let people live longer. Right now, we know they can't live without fermentation, and they can't burn ketones or fatty acids. I published a big paper showing all the cancer cells accumulate fatty acids in the cytoplasm because they can't use them. And if they try to burn them, they get reactive oxygen species and kill themselves. It's like a protection. Yet most of the people in the cancer field think that those fatty acids are there to drive the dysregulated cell growth. These people have no clue. They don't read the scientific literature, nor do they understand evolutionary biology. The fatty acids cannot be used as tumor cell energy, nor can ketone bodies. They're driven by fermentation. Fatty acids and ketones are non-fermentable fuels. Cancer cells thrive on fermentable fuels. What are they? The sugar glucose and the amino acid glutamine. Yes, we looked at other amino acids. We interrogated them all. Yes, asparagine can do a little bit. Glutamate can do a little bit, but not like glutamine. Glutamine is the big dog in the amino acids that can be fermented. So you have glucose fermentation and you have glutamine fermentation, and they can't use fatty acids and ketone bodies. So the solution to the cancer problem is quite simple. Lower the blood sugar, elevate the ketones, and then you come in with pulsing of the glutamine pathway, and you should be able to manage these cancers quite effectively without toxicity. And this is why we're seeing these parasite medications. They attack the same metabolic pathway that the tumor cell is dependent on. And they're very non-toxic, and they work together with the whole transition of the body away from sugars, away from carbohydrates, and elevating ketones. So this is a planned attack on cancer that will be strategic, based on evolutionary biology, and it's non-toxic. So the question you'd ask, well, how come all the big cancer centers don't know about this? And the answer is, I don't think they want to know about it.
Well,
and they're also indoctrinated to think it's a genetic disease. So how are you going to move the feet? You're not going to move the needle. Yeah,
although there are some doctors who will be frustrated in the lack of success. Although in my experience, I didn't want to go into oncology because I wanted most of the time to win. So there's a selection bias of the personality of someone who goes into oncology. But then the other phenomena that I notice is that if you're a researcher and you just get funded to study exercise, because that's what you're interested in, exercise, you'll never study diet, right? And so when Coca-Cola was funding an exercise researcher, he didn't care that it came from Coca-Cola. When what Coca-Cola was doing was keeping other people away from him, the researcher, right, to make him focus on something other than Coca-Cola. So the chemotherapy industry is going to keep these doctors busy, or radiation and chemo, the traditional tools. And if they fixed, 50% of people for a certain period of time, it seems some areas of oncology, there's been some victories, but there's a lot of change going on. I had a patient come to my office with a cancer, aggressive cancer, and I'm not known for treating cancer. It's more obesity and diabetes, but he learned about keto and cancer and had read your book and went to some other universities. And I thought, well, I'll just go to PubMed. I'll just see. I typed in the invasive thyroid carcinoma and then keto, and there was a paper that popped up from Matthew Nez, an EHS at Harvard, who was a surgeon who has an animal model. He came to this because his wife went to a keto meeting, maybe heard you speak, and then told, through pillow talk, that his husband started studying this, and it turned out that it had some benefit, reduced tumor growth in the animal model. So I'm hopeful that there will be people who will study this. And I just have to say, the one of the criticisms I got 25 years ago was studying a diet that was unusual and ostracized, was that a Dr. Atkins funded me that first study. And then I say through the years, the hallmark of science is replication. And so in your world, is there another center or investigator that has replicated what you've done? I think that's important.
I think the replications come here and there. Maggie and Brad Jones, who are doing the big documentary on the cancer revolution, showing that it's actually a mitochondrial metabolic disorder, are categorized or putting these folks into a group, all telling their story of how they use metabolic therapy to manage their so-called terminal stage 4 cancers. We're collecting a group of these folks, and everyone that I get, because I get a number of people emailing me who I send them my information, a kit of information, and they use it, and then you don't hear from them, and then all of a sudden, a year or 2, 3 years later, they come back and they say, I'm still alive. The doctors are always miffed. They have no clue what happened. And a lot of them don't even ask, what did you do? They just said, whatever you did, just keep doing it. They don't even want to know what the person did.
Is anyone helping you assemble these stories in a different year?
Yeah, I'm collecting. I have a registry now of folks, and then I share them with Maggie and Brad Jones. So Maggie and Brad then put these people into their documentary, and they have a paid. Now, it used to start off with a few of these people, and they were said, "Oh, they're flukes." They just, by the grace of God, something happened. Their tumor disappeared. Then all of a sudden, you get more of them, and then more of them, and more of them. And it's starting to be a stampede of flukes. So where the hell are all these? Why are these stage 4 terminal cancer patients still alive years after they were told that they put their life together and get ready for the end? And I find that when we get these people and they start the metabolic therapy right after their diagnosis, and they themselves are major participants in the management of their disorder, their condition, they are the ones that do the best. They are the ones that do the best. You the ones that feel, I don't want to do that. No, low carb, forget it. Those guys can go out and look for tombstones, because they're not going to be the survivors. The survivors are the ones that take an active role in the management of their disease. And I should also point out, Eric, that metabolic therapy does not guarantee cure to cancer. Metabolic therapy is a way to manage your disorder and keep you alive for a longer period of time with a higher quality of life. If you live to be 95 years old when you were diagnosed at 35 with a terminal cancer, and you die from heart disease at 95, and your cancer never came back, only then could someone would say that guy was. Pablo Kelly in the UK was diagnosed with a glioblastoma 10 years ago, and he's still alive today. Very funny, he was diagnosed with an inoperable glioblastoma, and that if he didn't do standard of care, he was not expected to live more than 9 or 10 months. He chose no radiation, no chemo, no steroids, nothing. The tumor continued to grow slowly. He had his first debulking surgery 3 years after his first diagnosis. Then he went on metabolic therapy. Again, another 3 years went by. He had another debulking surgery. It continues to grow. And then he just finished his third debulking surgery. No radiation, no chemo. It's funny. We were laughing the other day with Pablo. He says, yeah, I'm still alive over 10 years now with a glioblastoma. And they said it was inoperable. Now it's been operated on 3 times. But he's still alive. He got married. He has 2 kids. The issue here is he cured? No, he's not cured. Is he alive? Yes, he's alive. And he has a fairly good quality of life, other than a few seizures that he manages with ketogenic diet. So this is the situation.
So is that coincidence that you had a glioma model, and it seems like GBMs, glioblastoma multiformes, are somewhat, how would you call it, keto responsive, so you had an epilepsy and a glioma model.
Yeah. Well, I didn't have epilepsy. I had mice that had. I had 2 tumors that arose spontaneously in the brain of a mouse, and they were glioblastomas. And then I produced several by putting chemical carcinogens into the brain. So I produced a range. I produced a range of glioblastoma models that replicate the different kinds of cells, neoplastic cells that you would see in a human with GBM. So they're highly invasive mesenchymal cells, and then there are the rapidly proliferating stem cells. So you hear a lot about stem cell gliomas. You hear a lot about stem cells. Whatever the cell is, whether it's a stem cell biology or whether it's a mesenchymal biology, they're all dependent on fermentation for survival.
Is there a way so Dr. Nez at Harvard was explaining to me, just by emails, that some tumors are just very glucose dependent, and he is a surgeon who takes out these fast-growing tumors, but knows they're going to come back. And so can you test how responsive a tumor might be with that? Take a biopsy and put it on a petri dish. And what are your thoughts?
Well, what we found is that he's right. But the tumor, the human glioblastoma, is they used to call it. They don't use the term multiforme anymore. They just call it a glioblastoma. But the reason why they had multiforme, because it consisted of so many different types of cells in there. And you wouldn't know which part. Some people have a lot of what we call mesenchymal cells, which are a highly invasive part of the GBM. And others have a lot of stem cells, which are the most rapidly part, but they don't invade. They just rapidly grow. A lot of angiogenesis, a lot of blood vessels, inflammation, all this kind of stuff. And we parsed out these different kinds of cells. And we asked, some of them are indeed more glucose than glutamine dependent, and the mesenchymal cells are more glutamine than glucose dependent. So the way you kill them is you got to target both fuels simultaneously. Then you kill both kinds of tumor cells. Doesn't make any difference what they are. They're going to die because they need the fermentation fuels to survive. So that's why you target both together while transitioning the body over to ketones, because neither the stem cell nor the mesenchymal cell can burn fatty acids or ketone bodies. So we did all those studies, and we know now that how to kill these cancer cells is not that complicated. They are driven by a fermentation metabolism, and they can't switch to fatty acids or ketones, regardless of whether it's a mesenchymal cell, a stem cell, regardless of whether it's bladder cancer, colon cancer, breast cancer. They're all the same. They all are fermentable. It's a fermentation machine, just like Warburg said. Once you understand that, then you know how to go and strategically manage cancer without toxicity. The problem, Eric, is that I can speak until I'm purple in the face, and when I talk to some of these oncologists, it's like I'm talking to a plug socket. They just can't seem to understand what the hell I'm saying, and they don't read the scientific literature. They're indoctrinated to think cancer is a genetic disease, and the promise of the immune therapies, the genome projects, and all this stuff was to do precision medicine, target the gene, kill the tumor cell. That has not materialized because cancer is not a genetic disease. So we still poison and irradiate people. That was supposed to disappear when we realized the new precision medicine was coming. It's not coming because it's based on the wrong theory. So once you understand the theory, then you'll be able to manage cancer effectively without toxicity. The problem is I can't get that word. I publish in good journals. I give podcasts. I tell people I've got all these anecdotal evidence that it works. It does really well. And then people go to their oncologist and they say, oh, no, we have to use radiation and toxic poisons and all this other stuff. And we're going to use some CAR-T immunotherapy and Opdivo, bevacizumab, which is Avastin that should have been removed from the market. They're still using drugs that don't work, cost a lot of money, and hurt people. Now, what the hell is wrong with that? How doesn't anybody have a soul? Don't they understand what's going on here?
You know, I'll never forget a wonderful teacher in my experience was into did mismatched bone marrow transplants, which at that time, not many of them worked at all, and you would be watching people be very sick, and there was this saying or quotation. Desperate times, merit desperate measures. Well, so with the ongoing research, I'm optimistic about Jeff Volek at Ohio State being funded to do research. He had a paper recently on the feasibility of a low-carb keto diet for, I think, breast cancer, stage 4, or metastatic breast cancer. Do you think that just the keto diet itself, without the pulse therapy, is going to have the same possibility of effect, or is this going to be like the Women's Health Initiative study, $500 million? They studied the wrong diet. We don't want to study the wrong diet.
Well, I think there are some people who do in fact respond remarkably well just to metabolic therapy without drugs. What we think, though, is to get more people to respond. I think you're going to need some pulsing of the glutamine problem to get a number. There's several reasons. Number one, you can manage the cancer faster. You're going to kill tumor cells faster with press pulse. What we do in the world, where in fact we have a big paper that's under review right now on this whole thing. When the person is diagnosed, and that's regardless of what cancer they might be diagnosed with, we do a very comprehensive blood work to see how many blood parameters in all the panels, the lipid panels, the nutrient panels, the micronutrient panel, all these different panels. How far are they from normalcy? And generally, we put them on a low carbohydrate diet. It's much easier to jump into water-only fasting if you've been on a very low zero carb diet for a week or so. Then when you jump into water-only fasting, it's much less traumatic to the system. And then we start to see all the blood work starting to get into the right lanes, the right parameters, and then we monitor the blood glucose ketone index using the Keto Mojo meter, or whatever meter you can have, like sometimes Free Libra and all these different ways to measure blood sugar levels. And when you get into a low GKI of 2.0 or below, that's when we start coming in with the pulsing. That's when we hit them with the glutamine targeting drugs. Now these drugs work so much more powerfully when the patient is in low GKI, therapeutic ketosis, which is the biomarker, the physiological state of nutritional ketosis. And we're also realizing that even some of these poisonous toxic chemicals they're using on cancer patients can work so much better when the patient is in nutritional ketosis. So we're seeing that the dosages can be cut in half, 1/3 of what they used to give the patients. And still now the drug is doing what it's supposed to do without or minimal toxicity to the patient. Even radiation therapy, all the different, and then surgery, when like for glioblastoma, you can shrink these tumors down. And then when the surgeon comes in, they can debulk a much greater part of the tumor, which will allow the patients to live so much longer, rather than trying to go after the tumor when it's at its most aggressive state. Shrink it down, make it indolent, then debulk it. We have all of the tools needed for managing cancer effectively without toxicity available today as we speak. The problem is most folks just don't know how to use all the tools they have. They're not aware that this is a metabolic disorder and not a genetic disorder. And once they realize that, we're going to be able to manage cancer so much more effectively without the trauma and the toxicity and all the terrible things that we now associate with this disorder. It's just a matter of time once the people understand this.
So that kind of brings me to a practical matter in my office with my patients. I won't tell people that this guarantees anything. I don't think this guarantees to change your diet to a keto diet. It doesn't guarantee anything, but the studies ongoing, the idea that it's a metabolic disease, cancer is a metabolic disease. I still, the toxicities aside, I understand those, but I will still tell people to do what your cancer doctor recommends. But what you were just mentioning is using the diet as an adjuvant or as an added benefit or something to add to current treatments. And, and I just say this just because I have to play a little of the politics myself. If I tell someone that they shouldn't follow what their oncologist is recommending, I'm hearing what you say, though, that you may be able to get away with a similar medicine or treatment with fewer side effects at lower doses. But what I'll kind of say is that while there are no guarantees, this is something that I'll teach you how to do it and follow you over time. I'm just really hopeful because, of all the diseases, when Jeff Volek told me that he was going to study cancer, I was like, Jeff, of all the, if you did one big study on diabetes, we could turn the whole medical community, but he will have a lot of funding. Well, hopefully not too long. Hopefully he can crack this code at Ohio State. Thank you so much for your time. On a personal note, I think you met with my dad some years ago, and he finally passed in his early 90s. But thank you for all the help that you've given us. And the book, Cancer as a Metabolic Disease, is a must read. How do people find you today? Where would you like people to be directed?
Well, all of my research support now comes from private foundations and philanthropy. And people make donations to my research directly through the University Boston College. They just put the Seyfried's Cancer Research Program, but also Travis Christofferson's Foundation for Cancer Metabolic Approaches. He supports a lot of my research, a large amount of it. It's a 503 kind of foundation. So this keeps us going to keep testing it with the press pulse therapy. Dosage, timing, and scheduling is the will be like anything. Once you know how to use the tools that you have with respect to the dosage, timing, and scheduling, we're going to be able to manage this cancer quite effectively. People who want to do this, yes, Eric, it requires some participation on the part of the patient. They are key players in the ultimate management of their disorder. They are no longer a bystander. They are an active participant. And the more they understand the scientific concepts and what we're doing and why we're doing it, the more likely they will be the ones that survive really, really well from this treatment and this new strategy. Because, and one of the things we notice when you do metabolic therapy, we have many people who have diabetes, obesity, high blood pressure, hypertension. They have a whole range of different problems besides having cancer. And when they start managing their cancer, we see that the diabetes goes away, the high blood pressure goes away, the hypertension goes away. A lot of the chronic problems that they were suffering with, and they lose weight, they look healthier, they feel fit. So you're seeing a whole range of chronic problems go away when you're trying to manage your cancer.
Just maybe they're interrelated. Just maybe.
Yeah, they're very interrelated. And that I don't know is what people need to know that, though.
Yeah.
And once they realize that all these chronic diseases are interrelated, they might just embrace the whole thing and say, "What are we doing? How did we get?"
Well, you talk to a cardiologist; they only talk about the heart. You talk to a pulmonologist; it's just the lung. As a kidney doctor, we've subdivided the body up in so many ways. Thank you so much for your time, and just thank you for what you've done as a teacher and mentor to so many of us in the low carb world. Thanks, Dr.
Thank you very much, Eric. It was a real pleasure, and thank you for the interview.
Bye now.
Bye.
If you enjoyed this video, be sure to like, subscribe, and hit the notification bell, and check out adaptyourlifeacademy.com.